Okay. Okay, good morning. So our first speaker today, it's a real pleasure to introduce our first speaker today, who's Dr. Zoltán Molnár. He's a professor at Oxford University. Zoltán, you and I have known each other for like a long time, because he was doing a graduate work at the same time I was doing a graduate work, and we had overlapping interests. He originally thought he wanted to be a doctor and help people, but he decided to do science instead. He got his MD at the University of Szeged in Hungary and a PhD at Oxford University with Dr. Colin Blakemore. And Zoltán's work is really interesting. There are a number of people who look at the molecular development of the brain and how genes are deployed to generate aspects of cortical arealization in terms of different cortical fields and how they're interconnected and how thalamocortical afferents hook up to the cortex. But Zoltán does something that's really spectacularly different. He does comparative cortical development, and there are very, very few people who do this sorts of thing in both mammals and non-mammalian vertebrates such as turtles. So I looked at his CV. And I'm a number counter, many, many publications. He's on too many committees at the moment. This is the beginning of his sabbatical, so that's great. He has a number of awards, including the Circle of Willis, a Krieg Cortical Kudo Award, Rolliston Memorial Professor, is that correct?
Memorial Prize, yeah.
Memorial Prize. And I'm not going to make this too long. I'm going to let him get to his talk, but it's a real pleasure to introduce Zoltán Molnár. Thanks, Zoltán, for coming.
Thank you. I'm very grateful to Leah for all her support during the early stages of my career. You have been much more influential than you think. You know, when you start up in research, if you have a buddy somewhere in California or Australia, it's really helpful. So I really appreciate your help, Leah, over the years. So I would like to talk to you a little bit about comparative developmental biology, evolutionary developmental biology. And while we were gathering for this lecture, I showed you this video from Fatboy Slim, right here, right now. I don't want to play it again, but I just want to ask you, what mistakes did you notice in this video? What conceptual mistakes do you think are apparent in this video?
Not a single animal died.
Excellent. Yeah. So it was a continuum. You just had one single animal going, or human going. Other mistakes? You can see all the mistakes in the conception of evolutionary developmental biology in this single video. So I use it for teaching. Anybody else? Fine?
The conceptual mistake is assuming we want.
Yeah. Then bear with me for 3 more minutes. So let's try again then. So what do you think? What is wrong conceptually with this video? How is it portraying evolution? Sorry, exactly. Excellent. Scala natura. Number 2. Struggle a bit. Come on.
Nothing on variability.
Exactly. No variability. No selection. It's a single individual. No death. What else? So no population. Okay, before it's deteriorating any further, then I just show what are the main points I usually point out. So number 1 is that, exactly as you said, selection acts on the level of population. So when Darwin and Wallace, they started talking about natural selection, they were thinking of a population, not an individual. So they were thinking of a population of animals evolving over generations. So this was not really portrayed by Fatboy Slim video. The second is they didn't really show the diversification in the population because there was only one single individual in the video. So this was not really portrayed. And if you really want to tinker with these developmental mechanisms, you really have to see this diversification. So when you compare the hardware we have with a chimpanzee, and you want to understand how we have more cortical areas, larger brains, more cell types, and you probably know more about the cognitive abilities than I do, it's fascinating. And if you just look at the developmental mechanisms and the genes different between us, it's surprisingly little. So to understand this tinkering, I think we also have to understand these changes in developmental mechanisms. And even if you identify the genes, as in this case, they identified these networks, which are present in human but not in chimp, then basically you see that there are very few genes, and it's still very difficult to put your finger on the developmental mechanism which is producing this larger, more complex hardware, which is producing more cognitive abilities. So I think this is a real challenge for evolutionary developmental biology. Now the third mistake I would like to point out is that evolutionary change occurs not by the direct transformation of adult forms. You could see that the fish was transformed into lizard and so on within the life of a single individual. Of course, it will never happen like that. And in fact, when Haeckel demonstrated that there is a spectacular similarity at the early stages of embryonic development, and then they diverge. So initially you couldn't tell whether this will be a turtle, a chick, or pig, or human embryo. You will see this diversification throughout development. So he suggested that the early stages of development are more conserved than the later stages. And also, to some extent, the ontogenesis is a brief recapitulation of the phylogenesis. But in this statement, it is not true, of course, because you don't recapitulate all these. You have these trajectories which are similar. And probably you know the story. Haeckel was fired from his chair at Jena because he was drawing these embryos a little bit more similar than they were. So it was scientific forgery, and he was fired. Nevertheless, I want to point out that he was absolutely right. And if you have a look at these embryos in 3D rendering, initially you can't tell the difference, which one will be fish, pig, chick, or human. And however, at some point during development, you begin to see that this will be a chicken, fish, pig, and this is the human. So the initial trajectories of development are very similar, and then we evolved mechanisms to develop different organisms. And this is very much the same for the brain. So today we are going to talk a little bit about these evolutionary developmental mechanisms. And indeed, Haeckel was absolutely right. At early stages of development, you have more conservation. And if you look at the gene expression differences, at these early stages of development, you have a very nice correlation between different species, and then it will increase. And also, the age of the expressed genes are more ancient, and then you evolve different regulatory mechanisms later on. What is also interesting that it is not entirely true that the youngest are the most similar, the youngest stages, because even these very early stages evolved. But interestingly, you have here a bottleneck which all embryo will have to pass, and apparently this bottleneck is related to the formation of the different germ layers during gastrulation. And if you alter the basic interactions and you tinker too much, then you don't produce a viable embryo. So that's why these stages are very resistant for changes. It was Martin Raff at UCL who suggested that the embryo is just like medical students. So medical students usually don't know anything before or after exams. But during exam, they know how to pass, they memorize. And this is the same for the embryo. They have to pass this stage of gastrulation, and without getting this right, they can't continue into development. So that's why these early steps are so interesting. Another Raff, Rudolf Raff, was talking about the marriage between evolutionary developmental biology, which I shall mention a little bit later. So, and the fourth point to pick on the Fat Boy Slim video was that evolution is basically the evolution of development. And development was not really portrayed in that video. And Rudolf Raff suggested that between developmental biologists and evolutionary biologists, there was a very happy marriage about 150 years ago. But this somehow broke down. And in fact, we all work on a handful of species. Not here, not in this room, because you guys work on all sorts of creatures. But if you go to meetings, you will see that everybody's working on the same 5 model organisms. So the variety of the nervous system is not really studied to sufficient extent. And also development is also based on just a handful of species. And it's very, very difficult to work on other non-model organisms. So, and basically evolutionary biologists and developmental biologists got divorced. And now we are looking at these entities separately in different timescales, and this is wrong. I think we have to combine these studies to really understand how developmental mechanisms changed and evolved. And deliberately, that's why I gave the title today, The Evolution of Cortical Development. You probably know this extremely well, that nothing in biology makes sense except in the light of evolution. What I would like to push today is nothing in biology makes sense except in the light of development. And my graduate student, but now postdoc, Thomas Lickis said that nothing in biology makes sense. So in the development of the brain, there are 2 major steps. And during evolution, you had a huge radial expansion of the cerebral cortex. So initially, you just have a monolayer of cells. For instance, it's in the dorsal cortical area of a lizard or a turtle. We don't know what cells were represented here, whereas in mammals, you have a much thicker cortical layer, cortex with lots of different layers, and perhaps the supragranular layers increase the most. But this is not at all clear. So this is number 1. I shall be talking about how this hardware is becoming bigger during evolution. The second topic I shall be touching on is basically the tangential expansion, and Leah and John and others probably talked more about this, is the tangential expansion of the cerebral cortex. So you will have more and spread out in larger areas. So these are the changes in the hardware which will enable our brain to have higher cognitive abilities. And even this very basic principle, this radial and tangential expansion and the development of these is not fully understood at the moment. So what I'm going to do in today's seminar is, yeah.
Clarification. You emphasize the evolution of cortex, and then you say, and this allows the brain too. But I think you do agree, the brain comprises many other structures than cortex, right? So I assume you're not claiming that this change of functionality is only due to the evolution of cortex, or are you really claiming that?
You touched on a very, very important question. So if you have a look at a mouse where genetically you ablate all the connectivity between the cortex and subcortical regions, you can do that with a conditional knockout system. This mouse will run around, swim, even detect some visual stimuli. So there is no question that even with a servo isolé, you can move around in the environment, have locomotor functions and things. What I'm talking about is higher cognitive functions, speech, choreography, higher cognitive. You can't do that with your brainstem. So that's why we are asking these questions and not the reptiles. So we got this a bit better than them. So I think we do have a better cognitive hardware. I do want to make that point. It has to be the cerebral cortex, which is making us, enabled us. However, it is, of course, cannot happen just with the cortex. You have to have all the other subcortical structures. Look at the thalamus, for instance. Huge changes in the thalamus, especially the non-specific thalamic nuclei, pulvinar, huge changes. So it's very difficult to dissociate these, but you're right, I'm making a mistake. I always talk about cortex, but I shouldn't. So, nevertheless, most of the talk today, I shall be talking about cortex. And because we do believe that most of the higher cognitive functions are localized here. And I want to ask the first question that, is there an overall pattern of vertebrate brain organization in different species? So what bit is similar to what in a reptilian or a mammalian brain? How can we identify homologous structures or homologous circuits? Then I shall be talking about comparisons between mammals and avian brains, and especially I shall be talking about comparative transcriptomics when you compare the expressed genes in different parts of the avian and the mammalian brains. Then I shall be looking at what kind of developmental mechanisms evolved to generate more cortical cells on the larger cortical areas. So how could we tinker with the development to arrive to larger brains? And then I shall show some of the efforts we started making to understand lineage in different systems, in mammalian and reptilian and avian brains, to understand how these individual progenitors, suddenly they change their behavior and they are producing larger and more complex clones. Okay, so this is, I understand this is very much a point of view of a developmental neurobiologist. So I'm very happy if you interrupt during the talk and rectify some of the things I got wrong. So I'm interested in how these cells are generated in the right proportions and in the right numbers in different species. So if you look at the mammalian neocortex, then you see that the cells pattern into different layers. So you have different cell types, and also these cell types, they are combined in different proportions. And because of this, historically, the mammalian cerebral cortex have been divided up into 6 layers. But they could have done it, many other ways, 7, 8, or in some species, John will tell you, you have lots of sublayers in layer 3 or 4 in the primate. So, but nevertheless, we're now stuck with the 6-layered mammalian cerebral cortex. And if you look at these 6 layers, you have different cell types. For instance, layer 6 is projecting back to the thalamus, layer 5 to the spinal cord, basal pons, colliculi, and striatum, and other structures. And then you have cortical-cortical connectivity. So all these cells, they are present in different proportions, in different numbers, and they change these proportions in different cortical areas, which have been recognized by Brodmann, Economo, and others. So if you look at across the tangential domain of the mammalian cerebral cortex in adult, then you can see that different layers have different dominance. So for instance, in the primary sensory area, you have more layer 4 than in the primary motor, but in the primary motor you have more layer 5 than in the primary sensory. So these differences are very characteristic to different cortical areas because they perform different computational functions with different circuitry. And that's why these anatomical descriptions of these cortical areas have been so powerful in the past. And, yep.
For clarification, you just said that the number of layers is sort of almost arbitrary. It could have been 8 or 10, but it turned out to be 6. Is that really true? I mean, given that the program behind the patterning of a cortical sheet, you might not be able to generate a cortex with 20 layers because you lose control. You might not have enough different signaling systems to keep that stable, for instance. Is it really arbitrary?
So there is an overall pattern of cortical neurogenesis in all species. So there are similarities in the infragranular layer specification, and then there are specific programs for the supragranular layers. And this is very, very general across lots of different mammals, including human. What I was referring to is that you have, when you eyeball these patternings on these Nissl-Stein sections, you could have said, oh, why don't I put a border here as well? That you have 5A, 5B, you have, layer 4 has several sublayers, and should I really call it layer 4 of the upper layer of layer 4 or layer 3? So, but you are absolutely right. The general pattern, how these cells are produced, is highly conserved. So in the 60s, when this was described, you have an inside first, outside last neurogenetic gradient. So you produce the lower layers first, and then they bypass these, and then you have the subsequent layers generated. And this program, as you pointed out, is so general that if you isolate a fibroblast and you induce stem cells from this fibroblast, and you leave it in the dish, and you let it produce an organoid of a small brain, artificial brain, you still have the inside first, outside last pattern. And some of the neurogenetic programs are exactly the same. Of course, this is true for the early steps of development. Whether this is true for Alzheimer's, Parkinson's, or dementia, it's another question.
You still get the 6 layers in that?
You have to really push the gene expression to show that this is 6 layers, but you do have, so for instance, you have TBR1, FOXP2, ER81, ROR beta, ZB2. So these genes are roughly the same order in this organoid. So this is suggesting that somehow you have an innate neurogenic program which is producing this. What would be also interesting to do is to do the same from a turtle, reptilian brain, and have a look whether they can do that. Some people argue, Suzuki and others in Japan, that this very basic neurogenic program is present in the avian brain, and in some regions it's suppressed. And that's why you have just maybe supra or infragranular cell layers. But this is hotly debated, and it's not But nevertheless, so this is how we produce the brain. And what is interesting that in different mammalian species, you have lysencephalic and gyrencephalic brains scattered. So currently, we don't really understand whether brain folding was present at these very early stages, and it was suppressed in some species. That's what Wieland Hüttner and Bob Havner would like to suggest. Or maybe the folding evolved later independently in several species. So we don't know that. But let's just come back to the first question I wanted to answer and basically compare mammalian brains with sauropsids, so with reptiles and birds. If you look at a brain of a crocodile, let's start with the kangaroo, then you notice that it's very similar to other mammalian species. You have a nice, reputable cortex with 6 layers, and you don't have corpus callosum, for instance. You have a huge anterior commissure, but nevertheless, it's similar to other mammals. If you look at a turtle, however, you notice that there is a very thin dorsal cortex. This would be the lateral ventricle here. And then you have a bowl of cells protruding into the lateral ventricle, which is called the dorsal ventricular ridge. If you look at an iguana, it's even bigger. You see this bowl of cells. The lateral ventricle is almost completely obliterated here. And if you look at a crocodile, you see that this bowl of cells is even bigger. So my point is that the organization of this, of the mammalian and the reptilian and avian brain is very different. So they have a different strategy. They have different proportions in the brain to perform these functions. And this bowl of cells, the dorsoventricular ridge, is very prominent in these species. So the question, which was around for a long, long time, what is this dorsoventricular ridge? How does it relate to the mammalian brain organization? And there is no debate about the identity of this striatum. It is well accepted in both in mammalian and avian brains that these are the homologous structures. They have similar cell types, gene expression, connectivity, etc. Also, there is no debate about the homology of the hippocampal structures. People all agree, but we disagree what this dorsoventricular ridge, this bowl of cells, would be homologous to. So it is divided into further subtypes, subregions, mesopallium, nidopallium, and archipallium, and nobody agrees, whether they are homologous to the cortex or not. So a long, long time ago, in Leah's lab, with Paul Menger, we drove down some electrodes into the dorsoventricular ridge of an iguana, and we mapped the receptive field. And what was very interesting is that the receptive fields were very similar to a V1 in a ferret or a hamster or a primate within the dorsoventricular ridge. And also you had 2 visual representations with a mirror reversal within the dorsoventricular ridge. So if you just look at the receptive fields, you couldn't tell the difference whether you were in a mammalian visual cortex or in the dorsoventricular ridge. And Paul recorded from several species, so this was his opinion. But this is not a new idea. This was formulated long, long time ago by Harvey Carton, who, based on physiological properties, connectivity, and some gene expression pattern, already suggested that the dorsoventricular ridge, this bowl of cells in the avian brain, would have similar circuit elements to the mammalian cortical layers. So he based this argument on the thalamic targeting in the dorsoventricular ridge, and also with physiological properties and some limited gene expression. So according to Harvey, you would have an equivalent circuit from the thalamus, and the output layers would be also represented but not in layers, but as groups of cells. So instead of having these laminar structures, in the dorsoventricular ridge, you would have the thalamic recipient cells in one nucleus, then you would have the supragranular cells in a separate, and then the output would come from a separate layer. Whereas in the mammalian cortex, you have these separated into layers produced in the same part of the germinal zone in an inside first, outside last fashion. So this very influential hypothesis is called the equivalent circuit hypothesis, which dominated the field for decades. And you will see that it still has huge influence on the field. According to this idea, this hypothesis, during development and evolution, you would have a rearrangement of these cells in mammals. So you would have some kind of tangential migration from this zone of the brain, which was producing the dorsoventricular ridge in sauropsids, and these cells would migrate into the cortex to form these circuits. Of course, in these stages, this was not at all accepted by developmental biologists. The dominant idea was that most cortical cells, they arrive through radial migration from the local ventricular zone. So you can imagine how excited Harvey was when in '96, Juan de Carlos in Madrid started to put some tracers in the ganglionic eminence. So these are just bulges into the lateral ventricle below the cortex. And he noticed that some cells were leaving this zone and they were migrating tangentially into the dorsal cortex. However, it was very soon realized by several groups that these were tangentially migrating GABAergic cells and exclusively GABAergic cells. And also it was recognized that even in a bird and also in a turtle, you would have similar tangential migration where you wouldn't expect it. And you still have the same GABAergic cell neurogenesis here in this part of the brain, driven by DLX, LHX6, and other transcription factors. And this zone turned out to be different from the dorsoventricular ridge. So although there is a substantial component of tangential migration from this zone into the mammalian, avian, and reptilian brains, they arrive from a different part, and they have nothing to do with the dorsoventricular ridge homologous region segment. So now we also know that this is the same in human. Initially, there was a little bit of debate to what extent we can produce GABAergic interneurons locally. So in a primate, perhaps a larger proportion is generated here. However, this is also debated, and more recent work from the work of Arne Krickstein, for instance, is demonstrating that you have very similar gene expression in the ganglionic eminences in the human brain and also in the primate brain, and perhaps a large proportion of the GABAergic cells, similarly to the rodent, are generated here. So basically there's a little bit of debate on the proportions, but the general pattern is well accepted. So it has nothing to do with the dorsoventricular ridge, and these are only GABAergic cells. So, so what's happening then in the lateral, this stream of cells? So you have the GABA cells produced here in the lateral ganglionic eminence in all these species, but then you have a lateral stream which is not present in reptiles, and then you have accumulation of the cells in the dorsoventricular ridge protruding in the lateral ventricle, whereas in mammals you have some cells migrating to several other structures, so claustrum, endopiriform nucleus, lateral amygdala, for instance.
Something about the timing of this?
So these are all embryonic stages. So,
But since it's sequenced, right? So
Mhm.
it seems there's some also temporal patterning to this. And is the temporal patterning between mammals and reptiles comparable or different?
this is an extremely good question. When we want to compare time across different species, we are entering a very difficult territory. And Barbara Finley is perhaps the expert in this region. If you compare mammals, yes, you can possibly translate time, but you have to be very cautious because in some species, some parts of the brain mature faster than others. So, for instance, I always compare just the cortex, the timing, and that's relatively easy because you have a very general pattern. But if you look at the relative timing of development of the brainstem, for instance, or the thalamus, it can be very different. So, for instance, if you look at a marsupial, the cortex is really lagging behind, whereas the brainstem is way ahead. Just imagine you have to make a huge journey after delivery into the pouch. You have to crawl up. For that, you need a little bit of motor function from the brainstem. You probably don't need cortex for that. And then you just leech onto the nipple, and you stay there, and you develop further. So the brainstem is probably more advanced in a marsupial than in another mammal at these early stages, whereas the cortex is lagging behind. So to answer the question, what is the relative timing of these two streams, it would be very difficult to tell. But I think you touched on an interesting question. And the reason why I became interested in this part of the brain is because most of my research is actually on thalamocortical development. So I'm looking at how these cells interconnect with the cortex. And if you look at several mutants, this part of the brain is producing a major obstacle for the thalamic fibers. So and also the corticofugal projections. So the thalamic projections, when they reach here in certain mutants, they get deflected, they get default pathways down here, but they cannot cross this zone. And the same is true for the corticofugal projections. They get lost here. So ever since my graduate studies, I was very fascinated with this region, how these two sets of fibers approach and cross this zone. And we still believe that they cannot do that without one another. So they have to interact with one another, and we call this, with Colin Blakemore, the handshake hypothesis. So the two fiber systems have to interact. And I'm still working on this, on several conditional knockouts when you ablate certain molecules or cell types from the cortex or from the thalamus. And we still believe that this zone is, the timing is crucial for these fibers to pass. And perhaps the relative timing of this migration to this lateral stream and also the migration of the GABAergic cells may be related to all this. So if you really want to be frustrated in developmental biology, just study this region, because it's developmentally and evolutionary, it's perhaps the most complex part.
I'm wondering if there's a more fundamental question that needs to be addressed when I think about what the avian neurobiologists want to claim in terms of homology, which is the following. Even if all the cells, let's say you could explain all the cortical cells as having an equivalent in the DVR, isn't the fact that the cortex is fundamentally a differently organized structure than the DVR, which is maybe composed of nuclei, isn't that kind of the crux of the difference? It doesn't really matter what the origins, they can have a common origin, but if you're arranging yourself in different ways, it seems to be more computationally salient than just saying that they have a common developmental origin. Or you could say, or you could argue the following, which is, at some point if you go back far enough in development, you're going to find some commonalities across the two different taxa.
You just summarized what I wanted to talk in the next 15 minutes. Exactly. So if you already have that, then you can relax the next 15 minutes. This is exactly what I wanted to point, that we developmental neurobiologists, we attach a great deal of importance, what is homology? And our definition of homology is based on cell lineage. There's no question. You cannot argue that, it's the same connection, same gene expression. It is based on cell lineage. And you remember, John, when we talked in Budapest, and Neurogenesis said that be careful how you use the term homology. There is only one meaning of homology. And he's right. If you are a developmental neurobiologist, it's whether you come from the same progenitor pool, same segment of the neuroepithelium. But you will see that this is exactly what I will end up with after I take you through a long road looking at the comparative transcriptomics. But before that, I just wanted to point out that this rearrangement, so that you have a bowl of cells here, rather than this lateral stream and producing these other structures, can be reinstated in a mammal just by knocking out a single gene. So in a PAX6 mutant, if you look at this stream, this is now altered, and then you will end up with this bowl of cells. I'm not saying that PAX6 is responsible for this alteration, but I'm just saying that you can see this in this PAX6 mutant, and also you can see similar patterns in the PAX6 conditional knockout. So this idea, whether you have a nuclear to...
Are you saying that the PAX6 mutant doesn't have 6 cycles?
No, it does have alterations, but unfortunately this is such an important master gene that it's also involved in neurogenesis. So this system is not clear just to look at this. So what we would love to have is a mutant where you can only influence this stream, but not the cortex. Because that would be, of course, supportive for this nuclear to layered hypothesis. So this is one idea, which is a more colorful version of Harvey Carton's equivalent circuit diagram. And this is the other one, the nuclear to claustrum amygdala hypothesis, according to which this lateral stream has nothing to do with the dorsal cortex. It is more with the claustrum, amygdala, and the endopiriform cortex. It would be homologous developmentally. So I was fascinated with this question ever since I learned about this. And in fact, John Kars had a review in Teens in 95, which really helped me to understand these basic questions. And I wanted to understand this ever since. So if you are at home in the mammalian cortical cell type specification, gene expression, then you probably read these 2 papers where, so one of them is from Hirata's group and the other one is from Rexdale group, where they did the obvious experiment. So now we identified lots of markers for different infragranular cells in the cortex, so layer 5-6, and also some supragranular markers. And they are extremely good in delineating these cell types in different mammalian species. So what they did was they cloned up the equivalent genes and probes, and they looked at the gene expression in the chick and also in the turtle. And what was interesting is that the infragranular cells and the supragranular cells, they were separated according to the very similar pattern as Harvey Carton suggested. So you would have the infragranular cells, the projection neurons here, and for instance, the thalamic recipient cells in the dorsoventricular ridge. So these genes they selected, they are excellent markers for these cell types, and I would have selected exactly the same genes. I have to emphasize that these are extremely good markers. And the other publication also, they selected, for instance, ROR beta for layer 4 marker. So Jennifer selected this, and indeed this revealed these thalamic recipient cells in the chick brain, and then the output layers were also revealed with other markers which I would have selected. But the question is it enough to select 3 layer-specific genes, or do you have to select 6, 600, 6,000? When can you say that these cells are actually homologous or similar? So if you look at and you start comparing these equivalent circuits, then you are entering a really interesting field. Jarvis and others, they start talking about avian columns in this dorsoventricular ridge, for instance. So they say that these circuits, they are very, very similar to mammalian circuit where you have these columns lined up, but the origin of these cells is somewhat different. And I would like to say that for a developmental neurobiologist, it's completely different. It is indeed, these columns, they have nothing to do with one another. Even in mammals, to define a column is very problematic, but let's just gloss over that. But for a developmental neurobiologist, a column is coming from a progenitor from the ventricular zone, and then all the subsequent cells, so infragranular, supragranular, they are all generated from this zone in a sequential fashion. So this is what's happening in mammals. So these cells are produced in the ventricular zone. Whereas if you look at the avian and reptilian brains, what they are suggesting is that you have these different cell classes generated in different segments of the ventricular zone, and then you have the column perpendicular to that. So for me, this is not a column, developmentally speaking. They might have similar circuits, but for a developmental neurobiologist, this is a completely different strategy. So because of this, I really wanted to compare the mammalian and the avian brain in a more objective fashion. And the reason why we love to look at gene expression pattern and transcriptome is because it's quantitative. In anatomy, we do count cells, but until very recently, we didn't have really rigorous quantitative methods to quantify cells and cell types. Now, and probably John talked about this, there are some methods. But gene expression is easier to quantify and also to analyze computationally. So if you look at the adult gene expression pattern across the central nervous system, you see that the telencephalic vesicles will have certain genes are expressed, diencephalometer, and they preserve this gene expression in many different species. And it's quite useful for us to understand what part is belonging where. So also, if you look at gene network expressions, they are also extremely helpful to understand homologies. So, in fact, sometimes gene expression is used to look at relationship between different species to understand possible relationships. So that's what I really wanted to do with the mammalian and the avian cortex about 10 years ago. We selected the transcriptome. You could distinguish these cell types based on their developmental history, so lineage, anatomy, somatodendritic morphology, electrophysiological properties, etc. But you can also compare these cells based on the genes they express during development or in the adult. So we decided to look at the transcriptome of the supra and infragranular cell layers and have a look at it in mammals and also in birds. So for those of you who don't know what transcriptome is, it's basically the complete set of RNAs in a piece of tissue or cells. It is spatially and temporally organized. So if you take a tissue piece in different times of development or adult, it can change. Maybe it's even changing during the daily rhythm. And basically the transcriptome has lots of different elements. And I just put this on to remind us that you have lots of other RNAs, but I shall be talking mostly about messenger RNAs and long intergenic non-coding RNAs today. So you can look at these gene expression patterns with microarrays, and Allen Brain Institute had a really important role in doing that in many different species. And you can also look at these with sequencing. And this is now the dominant technology. And the reason why you want to sequence rather than microarray, because then you are not restricted, you look at splice variants, and also you look at these long non-coding sequences. So we decided to do the sequencing with, in collaboration with NIH. So we have these joint NIH Oxford graduate students, and they are superb. Especially Grant, he's a genius. And he pushed this project with the supervision of Chris Ponting, who is one of the leading bioinformaticians in Oxford. So we decided to compare avian and mammalian brain transcriptome in these regions. And we just wanted to see that how similar these areas are if you look at thousands of genes rather than just looking at a handful of them, as the Suzuki and the Rexdale publications did. So we selected regions where there is no issue with homology, like hippocampus and striatum, but we also selected other regions. So this took us quite a long time. So I'm going to talk about 6 years' work or so. So I dissected these layers in several adult mice, and just based on the site architecture. And then we sequenced the transcriptome. And also we heavily used the Allen Brain Institute Atlas, so that's why we selected P56, which is their time point as well. And Grant, through machine learning paradigms, tripled the number of layer-specific genes we now know, and we also included splice variants and also long non-coding RNAs. So we now have lots of layer-specific markers, and you can see this here in this website. So if you ever work on cortical layers and you want to compare it with other species, for instance, I recommend that you go to this, which is much better than the Allen Brain Institute website, because it has, it's a sequenced website, so you also have the long non-coding RNAs, and also you have the splice variants. Of course, they have different resources than we have. So what was interesting is 2% of the genes account for 50% of the poly-RNA and 75% for 99.9%. So some genes are expressed more than others. And also, you have some examples for isoform switching across different layers, which you would have never picked up with just simply looking at microarray data. So microarray is still cheaper, but then you miss these. And you also miss this long intergenic non-coding RNAs, and some of them have very strong layer specificity. For instance, this example. So our samples included these as well. And what was interesting is that some diseases are specifically associated to certain layers. Of course, these diseases are not just cortical diseases, but it was interesting to see that they are associated with certain layers more than with others. Now, after this, we also moved on to other subcortical structures and dissected these, and including claustrum, lateral amygdala, etc. And then we compared the markers. So basically, we wanted to define top marker genes for each region. And then we also wanted to see whether they are expressed more likely than expected by chance in any particular region. So, for instance, would claustrum and lateral amygdala share gene expression more than, let's say, layer 5 or others? So for this, I just show you a couple of examples. For instance, this gene is heavily expressed in hippocampus, but not other structures. And this one in the striatum, pallial amygdala. So we kept validating these with in situ qPCR, and now we had thousands of highly expressed genes. So at some point you have to say that that's enough. So we decided that maybe about 5,130 we cut off. We don't study the others. So for all these regions we identified about 5,000 most highly expressed specific genes. And then we moved on to the avian brain, And now we had to dissect some of these other parts. And it's not so simple to get chicken brain in England. So, yeah.
For your specific question, that seems fine for comparing, sorting out the homologies and so forth. For figuring out what's special about the human brain, it could be that it's some gene that's only expressed a little bit, for example, and any expression might miss something important, right?
I completely agree. And probably these low-end genes are the most important, and perhaps also at a specific phase of development. So we are here comparing adult gene expression. So what you really have to do is to repeat this study in many different stages of development. And I completely agree. But in those years, we have only done it in the adult. And your point is extremely relevant because my group is very much interested in this transient cell zone, the so-called subplate. So Pasko Rakic, Carla Schatz, Patrick Kennard, Heiko Luhmann, and others had huge contribution to this field. So at the bottom of the cortex, you have some very early generated cells. And these cells are extremely important to lay down the connectivity in the to and from the cortex, and also integrate into the cortex and lay down the functional connections. And once they have done that, they disappear. So these are transient cells. They are only present during development. And we have been analyzing the transcriptome of these cells in various stages of development. And what was interesting is that we identified only 3 genes which were expressed from embryonic 15 to all the way to the adult. 3. So it's a highly dynamic gene expression. So it's very important what stages you compare. And this is actually, I'm taking out the strength of my conclusions here, but we had to start somewhere, so we did it in the adult to compare these cells. But, and what is even more frightening is if you don't do these comparisons in development, for instance in the subplate, then you miss the genes which have disease association. So if you have a look at the hit list of the susceptibility genes of autism and schizophrenia, you can have hundreds of these genes. You will see that these susceptibility genes are associated to certain cell types, and there is a very strong association to subplate. My group showed that. And you can only see this during development. You have questions, Tony, or you just hold the microphone? Good. Just threatening. Okay. Okay.
Look, I have the microphone. The question is, of these 5,130 genes that you identified, how many are actually uniquely expressed in the brain, and how many are also expressed in other parts of the body?
We only did the comparisons for these genes within the brain, but for some of the long non-coding RNAs, we actually deliberately wanted to look at what other structures express them. And you can see them in the pancreas, testicles, gut, some of them. So the brain is not using a unique set of genes, or maybe we have to identify them. So many of these are expressed throughout development in other structures. So
You
that's
found evidence for the common sense belief that men think with their testicles.
it's a big jump in conclusion, but
It's a big achievement also.
you should have warned me, Liad. Yeah, so let's have a look at the chick transcriptome now. And so we had to go to an organic chick farm, and we had to, this was the team who had to dissect about hundreds of chick brains. And I don't know whether you have seen how chicks are processed. No, you don't want to know, otherwise you become vegetarian, probably. But the first thing they do is they cut the head, and the head goes down the drain, and it's washed away. So all we have to put some buckets there, and then we dissected the brains after it was frozen in the corner of this unit. And what we wanted to do is to dissect these parts and have a look at the transcriptome, how it is related. So when we were dissecting these parts, we were actually concentrating on areas which were inside these boundaries. And then, when we got the data, then Grant Bellegarde and Chris Ponting, they looked at these comparisons. So they did some specificity comparisons, marker comparisons, network comparisons, and module preservation comparisons of these over 5,000 expressed genes. So we did the same type of specificity comparisons in the avian brain as in the mouse. And the first question was whether we can see some regions with this specificity. So we wanted to get markers for the hyperpolyum, mesonido, archipolyum, etc. And Grant noticed immediately that there was a topological dissimilarity between the mammalian and the avian transcriptome. So to me, it means that they are very different, how they express genes and how they relate to each other. We didn't have any problems finding markers for certain regions. So LN Brain Atlas has no avian section, so we had to do the in situs and the validation ourselves, but it was quite convincing that we could find some markers which were specific for certain parts of the avian brain, and we had the homologous genes for mammals. And then we started comparing these regions, and no surprise, the striatum was very similar to striatum for all the 5,000 genes, hippocampus. Interestingly, oligodendrocytes in mammalian and avian brains have very similar gene expression. And then the next similarity which came out, and it's really frightening, is that the layer 4 of the mammalian cortex was very similar to the nidopolyum, to the structure which Harvey Carton suggested that is similar to the layer 4 almost 50 years ago. So these bioinformaticians like Grant and Chris, they can do these network comparisons and module preservation comparisons. So basically they are looking at how genes vary together. So these are called weighted gene co-expression networks. And they did that for all these parts of the brain like the striatum and also for the hippocampus. And you can see that there is a huge overlap between the avian and the mammalian brain in this sense. So you just say that, oh, you only have 6 genes here, but this is a very considerable overlap in these networks, suggesting that there are homologous networks in the two. And then what was interesting is that within the DVR, you had this co-expression between layer 4 and part of the DVR. And I have to emphasize that for me this was a revelation, because if you see there is a strong gene expression in the nidopolyum and then in layer 4, and these genes come from regions which do not develop from the same part of the neuroepithelium. So they are not homologous. For a developmental neurobiologist, they come from very different parts of the brain. Nevertheless, there were these striking similarities in gene expression. So what you just said 15 minutes ago or so is absolutely true. It doesn't matter where they come from. They can acquire that network of gene expression. And that's why I think it is such an interesting topic, because you have a network of genes which are only expressed in thalamic recipient cells, and although they come from different parts of the neuroepithelium, they adopt similar functions. And indeed, one of these genes, ROR-beta, has been implicated in all sorts of developmental steps in the barrel cortex, for instance, by Jeff Maclis's group and others. So I have to emphasize that these parts, layer 4, and this nucleus in the DVR, they come from different parts of the neuroepithelium. Nevertheless, they acquired similar gene expression networks. So I hope in this part I showed you, which you actually formulated in a much more elegant way, that you have some conserved gene expression networks between the avian and mammalian brain. You can see examples for diverse features where you have different anatomy and connectivity and gene expression. But you also can see examples for convergent evolution. So you have these two cell groups, they both received thalamic input, and they express similar genes. So I still maintain that although they show similar columnar organization, the two, the avian and the mammalian brains have a different logic in producing these circuits, and developmentally they are very different. Nevertheless, to get some functional properties, they use similar genes. And that's why I think this was one of the thorniest questions in evolutionary developmental biology, to understand what is homologous between an avian and the mammalian brain, what are the networks. So that's why I think it was Northcutt who coded the thorniest question of developmental neurobiology. Unfortunately, PNAS didn't take this cover, but we use it for next time. So after this comparison, do you have any questions for this part before we now move to the evolution of neurogenesis?
I also have a question that is, what does it say in terms of functionality to have similarities among those areas in the brain, either it is in the avian or in the mammalian brain? So what is the genetic network has to do with functionality in terms of behavior?
We did not address this, but this is the question. Exactly. Why is it that you have to express a network of genes in thalamic recipient cells to be able to process that information, although they come from different parts of the neuroepithelium? And whether you have to have similar gene expression to be able to meaningfully interact with the periphery. So you have 30,000 genes. Why couldn't we do that function with some other sets of genes? So I think this is a fundamental issue. And I think now we have modern technology to dissect these circuits a bit better. I don't know whether Leah or John, you know, anybody is doing some transsynaptic tracing with viral vectors in avian or reptilian, no? Because that would be really interesting to see what is the first target, second, third of these. So dissect these circuits a bit more, because I think very few people work on this, and most of this was done in the 60s, 70s, and now it would be nice to do it with modern anatomical tracing methods.
So when you look at some of the network analysis for some of those genes, when you do like a network analysis and you look at some of these homologous genes, and I know you didn't do it in the embryonic stages, but would you guess that in order for these cells to move from one place to the other, even though they're starting, like you said, in different embryonic tissues, that they'd have some of the same motor proteins or adhesion molecules, et cetera, because they have to migrate in similar ways, even though if they're starting from different places?
But maybe I didn't emphasize it enough, that these are two separate cell populations. They come from very different parts of the neuroepithelium. They end up in different parts in layer 4 and in the DVR. Yes, they have to have some motor functions and recognition and so on, but they both get thalamic input and they both have the same network of gene expression to some extent. So they acquire this independently.
So you would expect maybe even different gene products are at work for those two different.
Yes, for me
files.
they are not homologous. So probably they were generated through different neurogenetic programs. So just to make it absolutely clear, what we showed that they come from different pools, yet they have similar gene expression, and there is no migration whatsoever. You know, from these clones, we have nothing. They don't interact with one another.
Do you think I mean, there is a difference. One, you have this little thin layer of layer 4 cells, and then the others, you have this big clump of tissue. So you're saying they're sort of functionally the same, processing thalamocortical inputs. Wouldn't that make a difference on how things are processed, even just the structure, how thick they are, how the density of the cells? And has anyone explored those differences?
Of course, Harvey explored it, and every 2-3 years he has a review of the same. And I begin to warm to that idea because I think what he's saying is that we should really just stop arguing about how these structures come about. That, let's just forget it that they are, they are not homologous developmentally. Mind you, he is never saying that. He's still arguing. So if he could just say that, look guys, you're right, they come from different progenitors, there is no tangential migration to rearrange it, you still get these cells, yet they have very similar properties, and indeed to process this input and establish these circuits, you use the same set of molecules. And I think it's fascinating, no? Why is it that the transcriptome of the supra and infragranular cells is similar to the mammalian, although they come from different parts? Why couldn't we sort this out with other set of transcriptomes? So I think it's something is being revealed here about the logic, how these circuits operate and evolve, but we don't yet understand that.
And also constraints.
Yes.
There must be constraints.
And basically, although for us it's a very important question how these columns are formed in a mammalian brain, how many times these progenitors divide, how do we produce first these cells, then the others. If you just step back for a moment and you accept that you have different developmental mechanisms, and you just purely look at the circuits and you look at the similarities, I think that would be very interesting to do some comparative circuit analysis. And I don't think it's done right now.
So isn't there a hidden assumption in this observation? You're saying, look, the original location is different, but they end up in comparable locations with comparable functions, right? This is sort of the contrast you point out.
Mhm.
But on the other hand, as you indicated earlier, we cannot really compare these two embryos in terms of the temporal progression of their development. So in that sense, we look at location at a certain point in time of neurogenesis. And since we cannot answer the time issue, we don't actually know whether this is really a different location or not, because at an earlier stage of neurogenesis of this mammalian brain, you might have these progenitor cells in comparable locations, but that's not the point in time which you start to migrate, for instance. See what I'm saying? So the issue of
And
location
I'm going to show
cannot
you
be
the next
decoupled from
15
time.
minutes that that's not the case.
All right, great.
Exactly. So basically, at this point, what you really want to know is to go in here and understand progenitors, division of progenitors, migration of cells, to exclude the possibility that there is any mixing, no? Because that's what you want to do to really compare this. And what would be really nice to do is clonal analysis. So if you trace the derivatives of these progenitors here and you understand what kind of cells they produce over the course of development. So this comparative clonal analysis has not been done. And now the methods are here. And in fact, what I would like to do the next 15 minutes is discuss some comparative analysis of neurogenesis and also some methods how you can analyze clonally. And to take, to kind of summarize it already, I will show you that there was a huge elaboration of the germinal zone in mammals. So we have different progenitors in different properties, and they can produce more cells and with larger variety. So that will be my number one take on. And then I shall show you some clonal analysis showing that there were some intermediate progenitors evolved to make the brain bigger, and these intermediate progenitors contribute to all layers. And I will also show you that in the avian and the reptilian brain, you have just very limited neurogenesis closer to the ventricular zone, and you have no or very little intermediate progenitor pool.
So then, not to take too much in the next 15 minutes, but does that mean that the mammalian neocortex is more dynamic and more, I guess, changeable?
So what is fascinating about the logic of the development of the mammalian cortex, and I think it is unique for the cortex, is that how changeable it is. So basically, you evolved a mechanism which is producing a sheet of neuroepithelium where you have elements, circuit elements, which can interact with the environment. But the brain is not finishing the construction of this sheet of neuroepithelium. You have to interact with the environment from very early stages, including intrauterine life, spontaneous, sensory-driven, and subsequently sensory-driven interactions. And it's a very costly and long way of producing a nervous system, but it's adapting to the environment. So you have a huge scope to grow into that environment. And I don't think that we have that in an insect brain, for instance, or in other species. Maybe we have it in the sauropsids, but what is unique about the mammalian brain is that it's evolved to be able to evolve and develop further, more than any other structures, no? But this is making us also very susceptible for developmental brain abnormalities. If you look at the numbers, what kind of cognitive abnormalities we can have, it's, the numbers are staggering. So schizophrenia, 1 in 100, attention deficit hyperactivity disorder, depending on the statistics you read. Of course, dyslexia, 1 in 10 in the population, in the Anglo-Saxon world. So all these cognitive conditions or abnormalities are probably linked to this vulnerability because we leave some of these specifications to a later stage, and you have a very complex interaction between an unfolding genetic program, which would go on anyway, and the environment. And I think that's why I'm fascinated with the development of the cortex, because I think all these differences are imposed by the environment. I would not believe that you have a pre-specification here for a language area or for a higher visual area. It's driven by the sensory periphery and the interaction between the spontaneous and the sensory-driven activities. So I think you touched on an important point, that maybe the reason why we have this very elaborate, time-consuming way of producing a structure is to have huge flexibility and to grow this structure according to our needs. When you produce a computer, you generate all the boards, you wire it up, then you buy your program, and you switch it on, and it's working. No? That's how you do a computer. Our brain is switched on while it's being constructed, and we reinforce elements which are needed. And sometimes you need elements transiently, like I talked to you about these transient cells, the subplate cells. And once the construction is finished, you eliminate them. And without studying those steps of development, you would never understand how this complex circuit was developing. So I think that's why it's quite important to look at development and the stages. And in the next 15 minutes, I would like to convince you that there is no migration from here to here. and there is a huge explosion of neurogenesis in the mammalian ventricular subventricular zone. So we evolved neurogenetic programs for these progenitors to produce bigger brains, to expand the brain radially and tangentially. So I'm moving now the evolution of cortical neurogenesis. So the question is, how do we produce a bigger and larger cortical surface? And you will see that the reptilian, avian, and mammalian brains, they have, and I shall be mostly concentrating on this segment of the neuroepithelium from now on, because so how is it that this part is only producing less cells than this one? If you look at the brain development at these embryonic stages. So you have a coronal section through the brain. You see the lateral ventricles. This will be the cortex ganglionic eminence. This will be the hippocampus later on. You see that, so this is the ventricular zone, and this is the pial surface. By convention, I shall always put the ventricular surface down here. So if you time-lapse this zone during embryonic development, embryonic 11, 12, the gestational period in mouse is about 19 days, you can see that most of the divisions occur here in the ventricular zone. And then, the nuclei descend into the ventricular zone, then they divide, and then they move up towards the pial surface, and they will form this first post-mitotic cell layer, which is called preplate. And some of these cells will become the subplate cells, these early generated, largely transient cells. And the rest of the brain will develop from that. So if you want to capture these dividing cells, you can use an antiphosphohistone antibody in dead animals, because sometimes you can't look at the kangaroo brain alive, so you just look at the fixed brain. You can see that most of the divisions occur here in the ventricular zone when this epitope is exposed, but you also have some obventricular divisions, also in the ganglionic eminence where the GABAergic cells are produced. And you can also see that some of these cells, they line up in a zone which is called subventricular zone. So Paško Rakic coded subventricular zone. He should have really coded supraventricular zone because it's above the ventricular zone. But now we stuck with this nomenclature, so I will be calling this a subventricular zone. Cell biologists argue that since the basal membrane is here, this is apical and this is basal. So this is horrible field to be in because, because you never know who is talking about what. So cell biologists, they call this differently from us. Because we always say, oh, the apical dendrite is pointing here. But for them, this is apical because of the position of the basal membrane. So on cell biological terms, that's apical. So what I want to make the point in the next 5 minutes or so is that this zone expanded enormously in mammals, and this is how we produce more cells. And the subventricular zone will contain some intermediate progenitors which will amplify the output from these. So you have these radially oriented progenitors which were originally called radioglia. Of course, these are not glial cells. We now know that they are radially oriented progenitors, but we still call them radioglia. And then they will produce neurons straight away through direct neurogenesis, or through these intermediate progenitors. And sometimes some of these radio progenitors, they don't touch the ventricular surface, and then they have a different name, so they are called outer radioglia, and they are huge in primate and human. And there are some other progenitors which have a slightly shorter process, and they are called the short progenitors. So Paško Rakic identified. So at the moment we have these 4 progenitors. And
Can you cover that again? You just said something that made me feel like I'm 10 years behind on mine, maybe more than 10 years behind on mine. So my textbook understanding is that there are radioglial cells and that the cortical progenitor cells migrate along the radioglial cells. And then what you just said, I think, suggests that my thinking is now incorrect. Is that?
no, actually what's interesting is that if I understand glia in Greek, it means support. So when we have cortical development meetings, we argue whether we should change this name or stick to it. And Paško Rakic argues that since glia means support, radioglia, it's still valid and it's still correct that some of these cells migrate through radially extended processes of these progenitors. So this is still correct that you have the radioglia processes and then you have the radial migration, The only issue is whether we still call them radioglia or we call them radially oriented progenitor cells.
And that's because eventually, after they're done supporting their role of migrating other neurons, they themselves become.
Some of them they become, so they produce the whole lineage, not just neuronal, but also glial lineage. So these are fundamental progenitors of the brain. Some people call them stem cells. I wouldn't like to call them because we don't have evidence that they can become anything, you know. But these are neuronal progenitor cells, and basically they can produce other progenitor cells, whatever you call them, intermediate progenitors or outer radial glia progenitors or short progenitors. And what is so important about these intermediate progenitors, which were discovered relatively late, is that they can divide again and again in the subventricular zone and doubling and quadrupling the number of cells in the brain. So that's why evolutionary developmental biology is so interested in these cells, because we believe that they have something to do with the radial and tangential expansion of the brain. And therefore the regulation of these intermediate progenitors might be the key to enlarging the brains. So I will be talking a little bit about these in different species, okay? But the take-on will be that you have more of these cells and you have more of these outer radial glia progenitors. So if you stain turtle brains for antiphosphohistone H3 epitope, you notice that the dividing cells are mostly here in the ventricular zone, whereas in chicken, you have some subventricular zone in the subpallium, but not in the pallium itself. And again, we have done this in several stages. What is interesting, if you look at marsupials like monodelphis and wallaby, and in fact I worked on monodelphis in Leah's lab, I will show you there are some experiments which we must do in monodelphis. If you look at the early stages, P10, so they are already born, you still only have ventricular zone divisions here, and it's not until P16 when you have some subventricular divisions. And the same is true for the wallaby. Initially you just have a ventricular zone, then you have these subventricular zone divisions. The equivalent stages in the rat, you would have lots of subventricular zone divisions. Now, if you look at the size of the monodelphis cortex, I think many of you will be surprised to see that you only have half the number of neurons in a unit column in the monodelphis than in a mouse. So they have a smaller brain with less neurons, although the size of the animal is very comparable. I think Leah is holding that particular animal. And so what we could see is that if you have smaller cortex, then you have less intermediate progenitors, and therefore they only produce smaller brains. But the lineage of this has never been done in monodelphis, so maybe, Leah, we should do that. And if you look at the transcription factors which are associated to that particular zone of the brain, you will see that there is one which was identified by John Rubinstein and Bulfone and others, and this particular slide is from Robert Hevner's group, and this is called TBR2, and this TBR2 is associated to these intermediate progenitor cells, but just in the cortex, not in the ganglionic eminence, for instance. If you have a mutation in this gene, the TBR2 gene, then in human, then you have a polymicrogyria syndrome, smaller brain, microcephaly. So if you have 2 standard deviations below average, then it's called microcephaly. And in these brains, you also don't have callosum, corpus callosum. So we don't know whether it's going through these mechanisms, you know, that you have less TBR2 intermediate progenitors in human, but I shall show you some mouse experiments which we did in the TBR2 knockout and the conditional knockout, where we show that in these mice you have smaller pool of intermediate progenitors, and then you have smaller brains. So Liz Robertson in Oxford is interested in TBR2 at earlier stages of development for implantation and others, and they produced a conditional knockout which we looked at with Matthias Grosser. And the brains are slightly smaller. They don't have TBR2 expression. And when you look at the dividing brains, you have less intermediate progenitors revealed with phosphohistone H3. You have normal ventricular zone gene expression, I just show you here PAC6, and you have a drastic reduction in subventricular zone associated genes like SWET1, COX2, and NeuroD. So maybe that's why you have smaller brains in these conditional knockouts. So if you look at the cell numbers, you have a drastic reduction in all cortical layers in the cell numbers. And so you have no intermediate progenitors or less, and then you have a smaller brain. So we believe that these intermediate progenitors, they contribute to the cortical thickness. And I wanted to point out that on those experiments in the conditional knockout, we found that all layers were proportionally reduced, not just the supragranular layers. And at the time when we published that paper, the dominant idea was that these intermediate progenitors, they only produce supragranular layers. And I have to confess that I myself love this idea about that the upper layers are produced, because evolutionarily it would have made more sense for me at the time. So these intermediate progenitors express several genes, like SWET1, COX2, TBR2, and others, and the same genes are usually only expressed in the upper layers. So this is how this idea came, that the intermediate progenitors produce upper layers. But based on the conditional knockout data, and we suggested that that they probably produce all layers. And it was Robert Hefner who looked at the TBR2 EGFP mouse and showed that the derivatives of these cells were in all layers. So we wanted to trace lineage to understand how the brain is expanding. And in the last 5-10 minutes, we have to stop. Okay. Would 3 minutes be okay?
Doing fine.
Okay. Sorry. I can summarize it real fast, but for a developmental neurobiologist, what I'm going to show, this is the hottest stuff on earth. So it's really the cutting edge, you know, how you understand lineage. And we are in Barcelona, so you recognize this chap? He was the manager of Barcelona before he moved to Munich, no? Pepe Guardiola. And we paid 20 euros to see the stadium with these two chaps 2 years ago on fence, before we were robbed. And so Fernando did most of the development of these clonal methods I will be talking about. And Navneet, who is now a junior group leader in Edinburgh, is responsible for the TBR2 project, applying some of these clonal analysis and some other methods. So basically, Fernando and Navneet, they realized that we really have to do clonal analysis to track these two questions: where and what proportion neurons born from TBR2 progenitors, and what is the role of the intermediate progenitors in evolution of the neocortex. So what Navneet did was he wanted to fate map these TBR2 intermediate progenitors, and you can express TBR2 just in these progenitors, CRE, based on the promoter of the TBR2, and then you can have a look at what cells become stained if you cross it to a stop-floxed reporter line. So basically, he analyzed the number of TBR2 derived cells, and you can see that from 25 to about 50%, they all go through the TBR2 progenitors. And this would fit extremely well with Amanda's quantification in the TBR2 knockout, where we had reduction in all layers very similar to these numbers. So that would explain why we have seen reduction in all layers in the TBR2. So they were very happy with these results. So then they wanted to develop clonal analysis from these cells, and Fernando, who is a human frontier science program fellow in my laboratory, decided to develop a method where you express Cre under a selective promoter, and then you start cutting out these top sequences from different fluorophores. And if you have lots of different fluorophores, then you can generate many different colors, as Yvette generated in the rainbow mouse. But in the rainbow mouse, this recombination is not related to lineage. What Fernando developed is lineage specific. So these numbers, these colors, they represent actually whether they are clonally related or not. So this is a huge advancement in the stage. So just imagine when you go to your supermarket and you buy a bag of potatoes, then you go to pay, and then they just scan the barcode, and then ding, the price will come up, and then you pay for it. And if you buy 5 sacks of potatoes, it will happen again and again. Very reliable. Now just imagine if you have a combination of different colors, and you scan them in, and then you say, oh, this guy is related to this one, because they have that incredibly specific bar combination of colors. So this is what Fernando developed, and these random hues are present in these clones. And just to be on the safe side, he also drove some of this expression to the cytoplasm, nucleus, or the membrane, so increasing the probability so that we can distinguish them. And this is working extremely well. You can see some glial clones here, neuronal clones. In fact, glial clones are easier to detect because they have bigger arbors and easier to see, but the neuronal clones are also here. So now Navneet and Fernando use this to look at the TBR2 clones. So you want to know how many cells these intermediate progenitors will produce. So with this method, you can now detect individual clones rather than this mass production and just say that 25 and 50% of the cells come through them. So this is now working, and we can analyze these individual clones, and I can show you that we can now tell you about the clone size and also the dispersion of these cells. And what is very interesting, that these methods can be now used in reptilian, avian, and mammalian brains, so they are very happy with this. And we started to use these in chick, for instance. Obviously, in chick, you don't have Cre expression in the TBR2 cells, so you had to develop some methods to drive Cre expression according to some endogenous promoters. So Fernando did that, and this step allowed us to target the expression of Cre to different segments of the neuroepithelium, and then you can be absolutely sure that that clone was coming from here and not from here, which is a big issue. We discussed this with the DVR development. So now we are in a position to look at clones in chick and turtle, and you have to electroporate these in ovo, and then you can restrict the expression to different segments of the neuroepithelium, and we are just about to do this in turtle, but it's difficult to get turtle eggs, so we might have to go to Shanghai because they have more stages and better supply over the years. So the question will be, when we look at clones, clonal size, and intermediate progenitors, so we can identify exactly which developmental mechanisms change for what cell groups in the dorsal pallium, and what kind of mechanisms regulate this. So basically, this will help us to relate the equivalent segments of the neuroepithelium and see how they diverged to producing different cell types. But what some of you just said maybe an hour ago is the question. Then what do you do with these cells after? And how do you wire them up and use them for cognitive functions? And I did, as a developmental neurobiologist, I'm very interested in these questions, but it will take a bit longer. So what I showed you today is that you have, if you just look at the development of the forebrain, you have lots of examples for innovation, conservation, and convergence in the development. So you have examples for all these. I also emphasize the role of the intermediate progenitors, which I believe this is a truly mammalian innovation to produce bigger brains in the cortex. George Streeter would like to argue that you have a similar mechanism through intermediate progenitors in larger bird brains as well. So that will be interesting to see the lineage. But I believe that the expansion of the mammalian cerebral cortex is probably related to these intermediate progenitors. And then finally, I showed you some methods we developed to look at lineage and clonal analysis, which will actually help us to understand the origin and elaboration of these neurogenic programs. So I just quickly say thanks to all these exceptional people who did the work. Navneet was responsible for the TBR2 lineage analysis. Fernando, he is phenomenal. He single-handedly developed this clone method, which we now use. And Amanda, Waity, and Juan, they were responsible for the comparative neurogenetic programs. And of course, all the bioinformatics was done by Chris Ponting and his team, and especially Grant Belgard, who helped, and I'm grateful for the funders and also for your attention.
Okay, well, thanks for a fantastic talk. I think we've got time for a few more questions. Okay, I'm going to kick off. A lot of this stuff is new to me, and I'm trying to simplify it and make sense from my perspective, which is how we build technologies which exploit these same kinds of self-assembly mechanisms that we see in the brain. So we have two ideas about how brains build themselves. One is this idea about cell lineages, and different cells migrate to different places and then build circuits and so on. And that's more historical. And then we've got this idea about gene networks, which assemble circuits in place. And of course, these two things are the same, but understanding them and how they relate is difficult. And as you described it, there are really two alternative ways of thinking about homology. One is about cell lineages, and the other is about gene expression. And sometimes they agree and sometimes they disagree, as in this situation with the DVR and the neocortex. So, but probably what we want to do is think about fundamentally gene expression. And we have some genes which presumably are setting out the overall plan of the brain in terms of gradients, and then locally that's promoting other clusters of genes to work to build specific circuits. And my reading of what you're saying about the DVR and the neocortex is that the system is more plastic than we might have previously supposed, because a gene network in a particular part of the brain can take the local building blocks and assemble them into functionally similar circuits, even though the cells that it's using have come from quite different places. So, I mean, is that at all accurate as a reading?
Tony, I think I wish I had taken notes, because this is exactly what's happening. So what is the challenge is to dissociate programs which are largely automated and self-driven from those programs which are specific for those particular circuits. And then within those sets of programs, you will have subprograms which are actually dependent from the sensory modality or function. So I think that one would be interesting. So you have some very strongly inherent automatic genetic programs to produce these cell types. Then they assemble into circuits, and regardless of the lineage, they share some of these. But then you have a set of genes which probably only get expressed if you get exposed to certain environmental influences. So I think to dissect these will be quite a challenge for developmental neurobiologists.
Just a quick follow-up to that. It reminds me of some really classic embryology experiments, and then also some work that was done in the 80s. So the classic ones are like you take maybe it was a presumptive retina cell, and you implant it in the stomach, and it becomes a stomach cell, basically. So the environment in which a cell raised clearly matters a lot. And then there was a bunch of stuff, I don't know how well it's held up, by O'Leary, I think, in the 80s or 90s, that showed you could take, it's been too long, I can't quite remember, you take maybe a somatosensory cell and you transplant it somewhere else, and it takes on that identity. So in some way, this seems like a continuation of these kind of classic works, where it's not just the lineage, but the program that you should express given the environment. Maybe some of that environment is structured by the kind of information that you're receiving in some way.
So exactly. So your first example is probably so you have a cell which is it's gone down on a differentiation route. Then you put them into another environment, and then they are able to change their fate. So that's on a single cell level. What Dennis O'Leary and Brett Slugger did is basically they dissected late embryonic cortical neuroepithelia, and they basically put them from visual cortex into somatosensory position. And these blocks then received thalamic input, and in the somatosensory area, they receive patterned activity from the whiskers, from the contralateral snout, and then they imposed patterning within that cortex, which normally would have never developed these clusterings. And that clearly demonstrated that the general building principle of that piece of cortex is very similar, and with the sensory environment, you can actually influence the development, and then you have this clustering. But this is quite a brute force experiment when you have to transplant these. I think Mrigan Kassir did also almost 20 years ago or more, a fantastic experiment where he actually rewired the subcortical circuits. So now you are feeding in through intact thalamic connectivity. You don't change anything on the thalamocortical level, but you feed in the sensory input to a different thalamic nucleus, and then the different modality-specific input will then carve out a different cortical circuit. So I think that's an ingenious way of demonstrating that the information we process is building our brain. And he showed it very elegantly in the long-range cortical-cortical connections, patchiness of this, you can impose this. So this is related to the example I gave you that, this computer was wired up and then it was switched on, whereas our brain is switched on while we are wiring it.
Just one PS about the SIR stuff. People often seem to take it as showing arbitrary plasticity. As far as I can tell, and I emailed them recently, nobody's ever done it outside of primary sensory areas. So you can do this crazy, interesting transplant experiment, but the genes that are expressed in those areas aren't all that different to start with. Nobody's ever shown that you can do it arbitrarily. So there are important constraints where the lineage stuff.
So I want to ask something about the dorsal ventricular ridge. So it's sort of interesting to see that the response properties, it's sort of comparable to the receptor fields you find in V1, the point you also emphasized, right? However, in terms of their overall structure, it's just a ball of cells sitting sort of in the center. So sharing response properties with the neocortex, which is at the outside. So couldn't you argue that then the main difference between this mammalian brain and this avian brain is the ability to actually avoid collisions in cell migration? Isn't that the key switch that makes it different?
I mean, I can only have some theological argument about this. That So if you look at the sheet of the neuroepithelium in the mammalian cortex, and you accept that you can allocate proportions to sensory or motor functions according to the need, you can rearrange that on a sheet probably better than within a bowl of cells. But I don't know whether that's the case. Whether if you look at a DVR plasticity experiment, can you move things, shift according to the need? Probably. But my argument would be that the mammalian cortex is probably a better design to allocate these areas according to the functional need, whereas in the bowl of cells you have more restriction. But this is just.
My point is much more mechanical in that sense. Just to say, to go from this avian brain to a mammalian brain, the one problem you must solve is to avoid collisions in cell migration, so that you can allow these different subpopulations of cells to migrate without collisions. So to avoid collisions in this migration pattern makes the big difference.
So maybe what you are saying is that the mammalian brain is more suited to start producing cells. Then you have a constant platform on which some transient connections can lay out some rough topography when the distances are minimal before you finish constructing the other cells. So in that sense, yes, the mammalian brain is more suited to that you have an intermediate step involving subplate and other regions at the bottom of the brain, while you still produce lots of cells, so you can start building the circuits while you still produce neurons. So that would probably give you more flexibility to produce a bigger brain within those time constraints. So that's an interesting thought.
Do you have any questions from students? Okay, everyone's still taking it in. I think we should have a break because, yeah, so we start again at midday at 12. So once again, can we thank Zoltán for a fantastic